Hello, I'm Becky Kaplan from the Ball Lab at Duke University, and today I'm going to tell you about a story we recently published showing that TGF-beta and steroid hormone signaling mediate daf-16 cell-nonautonomous effects on starvation-induced developmental arrest. So starting off with a little bit of background, the overarching question our lab is interested in is how do organisms regulate their development in response to nutritional status? To address this question, we use our favorite model system, C. elegans. In C. elegans, in the wild, they often experience periods of feast and famine. When food is plentiful, the worm is free to focus on growth and reproduction. However, when food is limiting, the balance has to shift to focus on survival. We're interested in identifying the genetic pathways that regulate this balance. Many of the genes that have been identified so far are conserved in humans, and disruptions in these pathways can often lead to various diseases, including cancer, where cell growth is unregulated, and various metabolic diseases where nutrient sensing is impaired. So some members of our lab are looking at the effects of nutrition transgenerationally or maternally, but today I'm going to focus on the more immediate effect of starvation in L1 larvae. So C. elegans hatches from an embryo into an L1 larvae, and if food is present, they then progress through the larval stages and become a reproductive adult. However, if no food is present when the worm hatches, it enters what we call L1 arrest. During L1 arrest, development is halted, and the worm becomes more resistant to various stresses. This arrest is reversible, such that once the worm finds food, it exits L1 arrest and is free to develop normally. How is L1 arrest regulated? Well, one of the key regulators is insulin-like signaling. In C. elegans, the insulin receptor has one homolog, and that's daf-2. In fed worms, daf-2 activates a conserved PI3 kinase signaling cascade to repress the forkhead FOXO transcription factor daf-16 by phosphorylating it. When phosphorylated, daf-16 is kept out of the nucleus, and so it can't act as a transcription factor, and the worm then develops. Conversely, in a starved worm, daf-2 activity is suppressed, and daf-16 is then free to move into the nucleus and promote L1 arrest. So to study this arrest, we use mutants in daf-2 and daf-16, which have opposing phenotypes. So daf-2 mutants are constitutively arrested and starvation resistant, while daf-16 mutants are arrest defective and starvation sensitive. In a starved daf-16 L1, the normal somatic developmental events that occur in a fed L1 occur in the same chronological order, but at a much slower rate. Such widespread effects on larval development indicate that daf-16 regulates extensive permissive control of this larval development. To study the site of action of daf-16, we use GFP-tagged daf-16 expressed behind various tissue-specific promoters in daf-16 null animals. So here we're looking at starvation survival during L1 arrest. So in wild-type worms, you can see the curve in black on the right, just as the control, and then the daf-16 null is in red on the left. And as I mentioned, the daf-16 mutants are pretty sensitive and die relatively quickly. And then you can see that expressing daf-16 behind various limited tissue-specific promoters, including neuronal, epidermal, and intestine, as well as the native promoter, significantly shifts these curves back towards the wild-type control. So expressing daf-16 in even a very limited set of tissues affects the entire animal's phenotype, suggesting that daf-16 may be acting cell-nonautonomously. The other relevant daf-16 phenotype is how it's arrest effective during starvation. One of the markers we use to examine this arrest is GFP expressed in the single post-embryonic mesoblast cell, or the M cell for short, seen over here. So in the wild-type worms, during starvation, this cell almost never divides, as you can see by the non-existent bar over here. So, but in daf-16 mutants, a significant proportion of the population shows M cell divisions. Using the same tissue-specific promoters as from the previous slide, we can again see similar results here, where expression in the epidermis, intestine, and neurons is able to significantly suppress the daf-16 effect, though we didn't see any rescue from muscle expression. I also looked at seam cell divisions and a motor neuron differentiation marker that I don't have time to go into, but they're in our paper if you're interested. So the conclusion then is that daf-16 can have effects in cells where it is not present. Since daf-16 is a transcription factor, the only way it can be affecting cells where it's not present is by regulating some sort of signaling pathway. So our question then becomes, what signaling pathways are mediating the non-autonomy of daf-16? So I've drawn out a brief schematic of the pathway as we knew it when we started this project, and our area of inquiry then is downstream of daf-16. Our method to address this question was to use RNA-seq to compare wild-type starved L1s to daf-16 starved L1s. Among the significantly different transcripts, we then looked for known signaling genes. The candidates we pulled out were components of TGF-beta and steroid hormone signaling. We then proceeded to test these candidates in our assays. So starting off with steroid hormone signaling, I've drawn a brief pathway for you here. So daf-36 and daf-9 are components of the pathway that manufacture dafraconic acid, which is the ligand for daf-12, which is a nuclear hormone receptor. So we're again looking at M cell divisions in starved L1s. Notice that wild type and all of the single mutants show no divisions except for daf-16. So wherein daf-16 is showing divisions in about a quarter of the population. So using these opposing phenotypes, we were able to do epistasis analysis by building double mutants. Looking at the double mutants, we can see that there's significant repression of the daf-16 phenotype when we combine daf-16 with a mutant of the steroid hormone signaling pathway. Moving on to our second candidate pathway, TGF-beta. So this is the double-1 TGF-beta instead of daf-7. So in our analyses, I'm going to use mutants of the TGF-beta ligand double-1 and then the downstream transcription factor SMA9. So again, looking at M cell divisions, similar to the steroid hormone pathway, we see significant repression of the daf-16 phenotype in the double mutants. To conclude these experiments, we then decided that this looks like daf-16 normally inhibits TGF-beta and steroid hormone signaling to promote L1 arrest, and that this inhibition is physiologically relevant. Because TGF-beta and steroid hormone signaling are required then for daf-16's inappropriate development, we hypothesized that normally these pathways promote L1 development. So to assess this hypothesis, we took synchronized L1s and allowed them to develop on food for 12 hours and then scored how many times that M cell had divided. So as you can see here, in wild-type worms, most of the worms have 2 or 3 M cell lineages at this time, lineage divisions at this time point. But the TGF-beta mutants have significantly fewer divisions, mostly around 1. Then looking at the steroid hormone signaling mutants, the effect's not quite as strong as for the TGF-beta mutants, but there's still a significant shift with the curves shifting to the left and growing significantly slower than wild type. Now I'm going to show you a brief complexity we discovered in this pathway. So looking at these daf-12 alleles, we didn't see any effect. So what's going on? Well, remember that daf-12 is a nuclear hormone receptor, and as such, it has a ligand binding domain. So this is my little schematic down here. So this is the ligand binding domain. So the alleles where we saw an effect are actually point mutants in that ligand binding domain, effectively creating a constitutively ligand unbound form of the protein, compared to the nulls where the protein's completely gone. So in our model, we explain this by suggesting that the ligand bound form and the ligand unbound form of the protein actually have opposing effects on development, such that when you delete the entire protein with the nulls, you delete both forms, and the net effect is then 0. Okay, so finally, I want to return to the starvation survival phenotype of daf-16. So one hypothesis for the reason that daf-16 mutants die so quickly in starvation is that they fail to arrest properly, and then the development they undergo uses up their limited resources and they die quickly. So I've just shown you that by making double mutants of daf-16 with components of either TGF-beta or steroid hormone signaling, we can almost completely suppress the arrest effective phenotype of daf-16. So if this phenotype was correct, then these double mutants should also suppress the starvation sensitivity of daf-16. However, as you can see in this graph, we do not observe this. So the dotted lines are all of the daf-16 mutants, the single as well as the doubles, and they all cluster together. So even though the double mutants suppress the arrest defect, they do not suppress the starvation sensitivity of daf-16. Also, this is the TGF-beta pathway, but we saw similar results for the steroid hormone signaling. To then summarize our results with a genetic model, we started off showing that daf-16 had cell-nonautonomous effects on starvation resistance and somatic development. Using RNA-seq, we identified TGF-beta and steroid hormone signaling as potential candidates downstream of daf-16. Upon testing, we found that these pathways mediated daf-16's effects on somatic development, but not its effects on starvation resistance. We also found that normally TGF-beta and steroid hormone signaling promote L1 development, although the ligand unbound form of daf-12, along with its cofactor DIN1, appear to repress development. Taking this genetic model and putting it into the anatomical context of the worm, we see insulin-like signaling having a role in the nervous system, the epidermis, and the intestine. We think that this allows for sensing of the internal nutrient status in the intestine and external nutrient status via the sensory neurons. The components of the TGF-beta and steroid hormone signaling are also present in each of these tissues where they can be regulated by daf-16. Future studies can focus on trying to identify these unknown effectors between daf-16 and its effects on starvation resistance. But overall, we want to conclude that daf-16 promotes L1 arrest by inhibiting pathways that normally promote development, and that daf-16's effects on starvation sensitivity are not due to that inappropriate development. So that brings me to my acknowledgement slide. I especially want to acknowledge Yu-Tao Chen, who started this project and then left it to me when he graduated. Also Ryan, of course, for his guidance. And Colin, Jim, and Brad also directly contributed to this project. The rest of my lab members for their support. The Sherwood Lab at Duke has given us access to reagents and equipment, of course, our funding. And if you want to hear more from our lab, please go see our posters. Thank you. Any questions?
So I had a question.
Yeah.
So when you did your RNA-seq analysis, you actually got daf-36 as well as daf-12, is that right?
Yes, we got daf-36, daf-12, and dbl-1.
And so how do you explain the difference in the ligand binding mutations versus the null with respect to the transcriptional readouts that you saw with the RNA-seq for daf-12?
Right. Well, I think it's the regulation's more about whether or not the ligand's present, not whether the daf-12 protein is present transcriptionally. I'm not really talking like, does that make sense? Like the receptor is still expressed, and then which activity it has is determined by whether or not the ligand is there.
I see. So you didn't see a difference in the expression of the receptor in your RNA-seq?
There was one, but it was the weaker result. The ligand was the stronger transcriptional change.
Okay.
Yes.
Hi, yes, you said that the TGF-beta pathways are not important for the starvation resistance. I'm just wondering what you know about how daf-16 or speculate about how daf-16 is causing starvation resistance.
Oh boy. So the question is, how is daf-16 causing starvation resistance? I don't think we really know. That's the question. There's basically, I think what we could do at this point is go back to our RNA-seq and try and pull out more transcripts. Like, there was more results than I talked about. I just focused on the ones that we actually followed up on, but there's more data that we can go back and mine.
Okay, so if there are no further questions, thanks a lot for a great talk.